Hi everyone. In the immunology subject, we started off discussing about antigen antibody reactions in the previous part. In that part, we had gone through the uses of antigen antibody reactions, the stages in the antigen antibody reactions, then the principles of antigen antibody reactions, the mechanisms of antigen antibody reactions, where majorly we discussed about the Mark lattice hypothesis and the Jones phenomena. Now, in this part, we are going to majorly concentrating on the types of antigen antibody reactions. So, there are different types of antigen antibody reactions which are going to be called as first one, precipitation reaction, agglutination reaction, complement fixation test, neutralization, and labeled antibody assays. You all have to remember one thing. What is an antigen antibody reaction or immunoassay? So let's just recollect it. These are nothing but the biomolecular interactions between an antigen and its specific antibody are going to be termed as antigen antibody reaction. So there will be a biomolecular interaction between these two. So those are being termed as antigen antibody reactions or immunoassays, which are going to be of different types. Okay, we'll discuss all these different types in the different parts. In this part, we are going to discuss majorly about precipitation reaction. So how can we define a precipitation reaction? When a soluble antigen reacts with its specific antibody in presence of an electrolyte, so here the electrolyte is NaCl, at an optimal pH, that is 7.4, and the temperature 37 degrees centigrade, then the antigen antibody complex forms an insoluble precipitate, and this reaction is called as precipitation. That means when a soluble antigen reacts with its specific antibody in presence of an electrolyte and an optimal temperature and pH, the antigen antibody forms an insoluble precipitate. This reaction is called as precipitation. When the precipitate remains suspended instead of sedimenting, that reaction is called as flocculation. We will see what are these things. Okay, flocculation is a type of precipitation only. The precipitation reaction may occur in liquid medium or in semi-solid medium like agar gels, agaros, or polyacrylamide gels. So let's see the mechanism of precipitation. So you may already known about this mechanism of Mark's lattice hypothesis, but still let's have a glance of it. Okay, so here in the Mark's hypothesis, we know that we are having a zone phenomena. So according to the Mark's view on lattice hypothesis still holds a good. Multivalent antigens combines with the bivalent antibodies in optimal proportions, and the free antigen binding sites in antibody and as well as antigen determinant groups remain available after initial combination of antigen and antibody, which enable the complexes to link up to form a large lattice in the zone of equivalence. Now, this you can see the pictures in the part 1 where I explained it in detail. Okay, here we have to go in detail about the applications and the types. So, just I'm going to have a very fast thing. So, this now what is happening in the zone of equivalence, the antigen antibody aggregates are formed in such a way that there will be a large three-dimensional lattice structure through FCFC portions are formed. Now, this lattice consists of alternating antigen and antibody molecules. When the size of lattice exceeds critical volume, they settle out of the solution obviously. That means when you are going to see the precipitate at the bottom means you are going to have the size of lattice is going to be of more. So in the zone of antibody excess, so here antibody excess, all the functional determinants of the antigen molecules are soon taken up with antibody, so that very little linking occur between the antigen antibody complexes. In the zone of antigen excess, this is the post zone, less precipitate forms because the inavailability of the antigen antibody complexes to link up together to form the complexes, which results in the failure to form the large aggregate or lattice. But at the zone of equivalence, we are going to have the more antigen antibody aggregates are going to form. So that's how we can see the precipitation at this stage of zone of equivalence. Okay, so this is a mechanism which followed by the precipitation reaction. Now coming to the applications of precipitation, what are the applications where we are using this precipitation reactions means? As we know that precipitations are very sensitive and can detect as little as 1 microgram protein antigen, the test may be done either as a qualitative or quantitative test. The quantitative test is much more widely used to detect the presence of antigen and is of considered value in. So, for example, this is going to be having the considered value in the test like identification of the bacteria, that is detection of a group specific polysaccharides of streptococcus, that is going to be called as Lanzafield's grouping, or even it is going to be used to identify the bacterial components. One is identification of the bacteria, and the second one is identification of bacterial components in the infected tissues like Bacillus anthracis. Example is Ascolis dermoprecipitate test. Then the detection of unknown antibody, the test like VDRL and the CAN test. And then what we call it as medicological identification of human blood or the seminal fluid, standardization of the toxins and antitoxins, and even for testing the food adulteration. So these are the few applications of the precipitation reactions. Then moving to the techniques or the types of precipitation reactions. There are three principal types of precipitation technique. Simple precipitation, remember, simple precipitation, gel diffusion test, immunoelectrophoresis, which is also going to be called as gel electrophoresis or immunoelectrophoresis. So how many types of precipitation reactions are there? Three types are there: simple precipitation, gel diffusion test, immunoelectrophoresis. Coming to the first one, simple precipitation reaction. So if you see this picture, they are very simple. That means adding of an antigen and antibody. Then if you leave it, if you are getting the precipitation, then we call it as a simple precipitation test. So here you can see a clear solution of an extract of the test antigen or the organism is layered slowly onto a clear antiserum in narrow test tube. So here you are going to have an antiserum, and in this you are going to have the addition of clear solution of antigen is added. So here is the green thing, and now slowly it is added, and after a period of incubation you are going to see some sort of a precipitation between the junction at which of antigen and antiserum are present. So now this a white ring of precipitate appears at the junction of these two fluids, that is antigen and the antiserum. And this kind of simple precipitation test is going to be examples: Ascolis thermoprecipitation test, test for C-reactive protein, and even the Lanzafield's method of grouping beta hemolytic streptococci. Okay, so there are the tests where we use this one. So the simple precipitation test can be of, again we'll classify into ring test where you will get the precipitation ring. So that's why it is going to be called as ring test. Then we can have the flocculation test. Okay, this is a tube test, and another one is the slide test. So let's see those. So we had already seen the ring test. What is happening? We had taken the standard clear anti-sera in the test tube, and then slowly we are going on adding the clear solution of an extract of the test antigen or organism, and slowly if you are going to layer, and then you are going to have a period of incubation where if they are going to be a specific with one another, you are going to have the formation of a white precipitin ring at the junction of these two liquids or solutions. So this is all about the first type of simple precipitation test, that is ring test. Then moving to the second test, that is flocculation test or the tube test. So what is happening here? The antigen solution and antiserum are taken in a test tube mixed. So you are going to take simultaneously the antigen as well as the antiserum, and they were mixed. So and incubated and then absorbed for the precipitation. Usually several tubes with different dilutions of antigen are set up since precipitation may be inhibited in the post zone. So that's the reason they are going to have the multiple of test tubes. So the best example of this flocculation test or the tube test is going to be CON test, mainly used for the diagnosis of syphilis. And even a quantitative tube flocculation test is used for the standardization of the toxins as well as the toxoids. So here you can see if they are going to be a specific with one another, that is antigen and antisera containing antibodies are specific, so you are going to have the floccules. So I told you the formation flocculation. So what is meant by flocculation? When the precipitate remains suspended instead of sedimenting, instead of getting here, if they are going to suspend it throughout the solution, we call that process as flocculation, and those particles are going to be called as floccules. Okay. So this is all the things that we have discussed. So example is the Kahn test. Then moving to the slide test. So obviously the name itself is indicating a slide test where we'll perform the test, precipitation test on a slide. So here also you are going to add a drop of antigen, maybe of VDRL antigen. Solution is added to a drop of decomplemented patient's serum on your slide, mixed well and shaken for a few minutes. So if you are going to have the visible clumps appear in possible cases, that is positive cases, then we consider that as a positive. Okay, so this is all about the simple precipitation test. So what are the simple precipitation test poses? Three types. One is a ring test, then we are going to have the flocculation test or the tube test, and then we are having the slide test. So the example for this slide test is the VDRL test or RPR test, rapid plasma reagent test, which is used to identify the syphilis disease. And then moving to the second type of precipitation test, that is gel diffusion test. So listen carefully. This gel diffusion test is going to be of very important in the exam point of view. So coming to the gel diffusion test, the precipitin test done in agar gel is called as gel diffusion or immunodiffusion. The gel diffusion test are also called as immunodiffusion test. And this diffusion test is going to be of giving more sensitive and specific results. And we can see the reaction is visible in the form of a distinct bands of precipitation, and reacting bands can be stained for better viewing as well as for preservation. So by this gel diffusion method, large number of antigens can be studied by this method. When only the antigen or antibody diffuses with the corresponding antigen or antibody incorporated in the agar, this is referred to as a single diffusion. That means this gel diffusion is again being classified into two types. How many types? Two types. One is the single diffusion. The second one is going to be the double diffusion. If only one, either the antigen or antibody is going to diffuse in the agar, then we call it as a single diffusion. If that is going to occur, that means when both the antigen and antibody diffuse, then we call it as a double diffusion. So different modifications of the diffusion techniques are in use, and several hours of diffusion are often necessary before the occurrence of precipitation. Remember, the gel diffusion test is also a type of precipitation test, but we are performing this in a base medium of agar or agarose gel. That's why we call this as gel diffusion test. Don't get confused that this is another type. So this is also a type of precipitation. Coming to the first type, that is single diffusion. So that means gel diffusion is going to be classified into two types. One is single diffusion, another one is a double diffusion. In the single diffusion, only one will diffuse, either the antigen or the antibody. Okay. So in the single diffusion, again it is going to be of two types, that is whether the diffusion is going to be in one direction or in the two directions. So here we are going to discuss about the single diffusion in one dimension or direction. The method that is going to be called as Hoarding method. So if you see the picture, then you'll get the clarity. So here is a agarose gel or agar gel which contains the antisera containing antibody. That means the soluble antigen is directly layered. So now the agar gel containing antisera is placed in a test tube. Now our soluble antigen solution is directly overlaid containing the that is antibody containing gel. Now what happens? The antigen is going to slowly diffuse into the gel and forms a line of precipitate or precipitate band at the junction of where the antigen and antibody is going to meet. So this is going to be of single diffusion in one dimension. Why? Only the antigen is moving, that too in one direction. This is the downward direction. So that's why this is going to be of called as single diffusion in one dimension. Then moving to the single diffusion in two dimensions. So this is going to be of example of this method is Manson method. So two dimensions or more than two dimensions, more directions. Only one is going to move. So you have to remember this one. Antigen is going to diffuse, but in two dimensions or more dimensions. The example of this method is Manson method. So what is happening here? The specific antibody or antisera is incorporated in the agar gel. So here you can see this is what? Okay, on your slide or petri dish, and antigens are placed in a well. So we have to create a well, and the antigens are going to be placed in that cuts or the wells. Now what happens? Slowly these antigens are going to diffuse into the medium or the gel. Now what happens once? They are going to have the formation of a precipitation ring around a well that contains the corresponding antigen. The higher the concentration of antigen, the larger the ring. So here it is going to have less concentration. So here the small ring. So here a medium size, and here you're having a large ring. Radial immunodiffusion is mainly used to detect and measure immunoglobulins in serum. So we can call this as a radial immunodiffusion, as they are going to be diffused in a radial manner. Okay, so that's why single radial immunodiffusion or Manson method. So where the position of the antibodies are going to have the connection with this antigens, if while they are diffusing, they will form the precipitate ring or the radial ring. So now moving to the next type. So this is all the explanation that I gave you. Okay, now coming to the double diffusion. So until now we have seen the single diffusion in one dimension, that is here it is one dimension, and here is a single diffusion in two dimensions. So here only one direction the antigens were moved, and here they are moving in two or more directions. So this is going to be a diffusion in two dimensions. Then moving to the double diffusion. So the name itself is saying that both are moving. Antigens as well as the antibodies both have to diffuse to form the precipitation band or the line. So double diffusion in one dimension, the method is going to be Oakley-Faltropy method. Double diffusion in two dimensions is Ochterlony double diffusion method. So let's begin. So in this technique, antigens and antibody diffuse towards each other, and where they meet in optimal proportions, a visible line of precipitate is formed. And the intensity of the thickness of the line of precipitate often reflects the semi-quantitative amounts of antigen and antibody that combine. So as we know, the several varieties are available: Oakley-Faltropy method and Ochterlony double diffusion method. So see the first one, double diffusion in one dimension, that is Oakley-Faltropy method. So here is a picture of it, then you can understand very clear. So here is the agar gel which containing the antisera in it, and you are going to have the place of plain agar. Here it is not going to have any antigen in it, just the plain agar was kept. Okay, then above which you are going to have the antigen solution is going to be placed. Now here the antigen have to diffuse, as well as the antibody also diffusing into the agar's medium or the plain agar. So at the region where the both are going to meet, you are going to have the precipitin band. So that kind of moving or diffusing of both the antigen and antibody is going to be double diffusion. As they are moving in the same direction, we call it as double diffusion in one dimension. So here you can see this method separates the antigen antibody solution with a layer of clear agar, and this results in the diffusion of both soluble antigen and antibody into the agar and the formation of the precipitation ring or the precipitation band. Then moving to the double diffusion in two dimensions, that is Ochterlony double diffusion method, which is very, very important. Okay. Next, here what is happening means, So we have to prepare what we call it as agar gel in a petri plate or a slide, and then uh, using a template we are supposed to make the wells in this agar gel. Antiserum is going to be placed in the central well here, okay, and in the surrounding wells you are going to place the antigens. Now what we have to do is we have to allow this plate for incubation, that means allow it for the diffusion purpose. After some time you're able to see some sort of precipitin bands develop where the antigen and the antibody is going to meet. Okay, so this is an antisera which is getting diffused and antigen is also going to diffuse. So both are diffusing and in more directions, that is more than two. So we call it as a double diffusion in two dimensions. Okay, now what is happening when the two antigens, that means when the two antigens, these two, or if it is going to specific, we are going to have the lines of identity for this antisera. The lines of precipitate fuse and is when in case of unrelated, that means this antisera is not related to these antigens, then it is going to be of unrelated like a cross each other. And there will be a formation of some sort of a spur formation. So here you can see which is indicating the partial identity. So that means it is going to be of relevant to this 2 1, but it is not relevant to the 1. So that's how we are going to have the three types of precipitin bands which we can be of these three different types of patterns. If we are going to have the formation of a band like this, we call it as these two antigens, that is 1 and 2 antigens are identical to this antisera. If we are having this cross like one, that means these antigens 1 and 2 are non-identical to this antisera. If you are having the partial like this, we call it as partially identical to the antisera, which we call it as a spur formation. And this technique was routinely used in the fast diagnosis of smallpox by testing the extracts of smallpox lesions against the antisera. And the precipitin bands appear within 2 to 6 hours. When the process is combined with electrophoresis, we call this as, that means if octolony double diffusion is going to be combined with electrophoresis, that technique is going to be called as counter immunoelectrophoresis, where the precipitin bands are going to be formed within 30 minutes. In general, by this method, it takes 2 to 6 hours. But if it is going to be a counter immunoelectrophoresis, it will appear. That means you are going to get the results within 30 minutes. So this is all about the double diffusion in two dimensions. So one more type of precipitation test is there, that is immuno electrophoresis or gel electrophoresis, that we will discuss in the next part. Okay, with this part we have finished the types of precipitations, that is a single diffusion where we had two types, then double diffusion where you are going to have the two types. So here it is a Occliffe-Althorpe method and this is a Octoloni double diffusion method. Thank you.